I think we're ready. Hello, everyone, and a warm welcome to you all to this webinar titled, Why is Trypan Blue Toxic and Unsafe to Use? My name is Janny Peterslund, and I'm Scientific Affairs Manager here at Chemometic, and I will be your host for this webinar. And we're really excited for today. We had about 350 people sign up, and I can see that there's still people trailing in, but we're at about 100 participants live now. So who are we at Chemometic? Well, we are a global leading manufacturer of automated cell counting and analysis instruments. We develop high precision equipment for consistent data delivery to thousands of customers in biotech, pharma, and the academic segments. And our automated cell counters are used primarily in the cell therapy, virus, and antibody production areas. The agenda today is that first, our speaker will give a 15 to 20 minute introduction, followed by a Q&A session of about 15 minutes. That's the right slide. Great. And if you have any questions, please feel free to submit them through the Q&A function of the Zoom in the bottom of the panel or the window that you're on. You can submit throughout or towards the end of the webinar, and then we'll ask them during the Q&A session. So if you have any technical question, you can use the chat function and tell us about those there. And just so you know, today's webinar is being recorded and will be shared with you afterwards. So I'm joined today by Dr. Torsten Schultz-Larsen, who is the lead application scientist here at Chemometic. Torsten has a PhD in molecular plant biology from the Sainsbury lab in the UK, and also has a postdoc at the University of Copenhagen. So Torsten, you'll tell us about how counting cells using trypan blue as a viability dye is maybe not the ideal way, and also present some alternatives.
Thanks for that kind presentation, Janny-Marie. And yes, we'll talk about trypan blue today. So the key takeaways from today is that you'll learn how does exposure to trypan blue affect your cell count. Specifically, how's the viability of your cell population underestimated? Will you see any viability change over time? And last, I'll touch on some safe alternatives out there. So with that, let's start at the beginning. And what is trypan blue? Well, it was developed in 1904 by Paul Ehrlich, who's a German physician, and later used by Edwin Goldman to describe in the seminal study the blood-brain barrier in mammals. And basically, see in the bottom picture, we see a rat injected with trypan blue, and in the panel on the right, we see that this is unable to penetrate into the central nervous system, therefore showing that you have a blood-brain barrier. Other things that trypan blue is used for, you have derivatives that are used to treat sleeping sickness. Today, it's mostly used as a viability dye. It's extensively used, actually. And how is it used? Well, trypan blue is used as it is unable to penetrate membranes, so it's a membrane impermeable. Therefore, living cells which have an intact membrane are unable to be stained by trypan blue. However, dead cells that have holes in their membrane will be able to be stained by trypan blue. This is called the dye exclusion principle. And in the three figures below, you can see how a microscope view of all cells would look like and how it would look with dead cells. You can segment those. And then from those, you can say, well, once I count all cells and dead cells, I can estimate my viability. In this example, it would be 10 cells minus 4 divided by 10, meaning you have a viability of 60% in this case. So how can you dye? Well, there are many ways for cells to dye. There are some that I like to call artificial, and these include fixation, for example, with ethanol or DMSO or NBF. Others include temperature-induced killing, and that can be heat treating. It can also, to some extent, be by cryopreservation in some cases. And likewise, you can use osmotic stress to artificially kill your cells. In contrast to this, there are some things I like to call natural ways for cells to die. One of these being drug-induced, for example, by camptothecin, where you'll actually use the cell's own machinery to execute cell death. You can also have that during differentiation of cells, for example, iPSCs to retinal pigment endothelial cells. Likewise, you can starve your cells and kill them that way around. So there are different ways for cells to die, and not every way of dying will give you the same result. So one way for cells to die is shown in this paper by Chan et al., where they basically took Jurkat cells and grew those in the lab, and then took out an aliquot from the incubator and left those overnight on the lab bench. And what we see here is that when you treat these cells with trypan blue, as we see in the top left panel, we see that the dead cells will actually start to swell. How are we sure that these are actually cells? Well, at the same time, these cells were labeled with fluorescent, the DNA binding dye, propydium iodide. And we see a nice co-labeling of these. So basically, trypan blue will in this case cause cells to swell and in some cases rupture. Likewise, you can ask what will happen with other dyes, fluorescent dyes. In this case, they've shown propydium iodide, and they show that this does not have the same effect in the cell. So really showing this is an effect of the trypan blue salt. So what is this caused by? Well, they show that it is actually cells, and trypan blue really causes the cell to swell. So they did a study where they then loaded cells into a slide and added trypan blue to one end of the slide, and then watched as the trypan blue diffused through the sample and imaged, in this case, one cell we've shown here. And we see that prolonged exposure will then lead to a swelling of the cell at the right. So basically, they swell. And what is this caused by? Well, it's presumably caused by osmotic pressure. In the top panel, we see a quarter strength PBS where we have a lot of diffuse objects that are harder to identify as cells, at least compared to the live cells. And then on the right panel, you have a 10 times strength PBS where we see a nicely defined cell and obviously have a much higher osmotic pressure in the sample. And if you quantify this, then you see that the intensity increases from low strength PBS to high strength PBS. So basically, trypan blue probably exerts this effect through osmotic pressure. Then you can ask yourself, are there other things that you should consider when you use trypan blue? Well, one thing you should also consider is that trypan blue exposure will alter the viability of your cells over time. So this is just one example by Soros et al. From 2013, where they've taken human trabeculate cells and cultured them for a day, and then exposed them for an increasing amount of time to trypan blue, and where they see that, granted, after an hour's exposure to trypan blue, you'll have a dramatically reduced viability of your cell count. So it is something to take note of, that if you use trypan blue, you should try to count your cells fairly rapidly, as they will start become labeled with trypan blue in any event. So really, what can we conclude on trypan blue based cell counting? Well, trypan blue causes swelling of dead cells, and this is presumably due to the osmotic pressure. This will lead to, unless you take it into account, that you'll miscount the number of dead cells and thereby impact your viability reading, and your total cell count will be slightly lower and your viability will be slightly higher. Obviously, it will not affect your live cell count. Likewise, trypan blue negatively affects cellular viability over time. So it is fairly important that you count your cells quickly. And then it's time for a short poll that will be shown on your screen now. And I'll just give you, like, let's say, 30 seconds to fill it out, and then I'll continue. Right, so I guess that most of you have had time to fill out the poll now, and I'll just continue, but do feel free to fill out the poll in your own time. So what about the toxicity of trypan blue? Well, according to the European Chemical Agency, trypan blue is a substance that may cause cancer or genetic defects or damage to fertility or the unborn child. And with this classification, what they're really trying to do is to nudge users to find other alternatives, or to the very least create safe workflows. So one example from Denmark is that you're here, you need to look for suitable alternatives, and if you're unable to find that, you can apply for a permit in order to be allowed to work with trypan blue. And really what you need to do as well is you need to create safe workflows, standard operating procedures for your lab professionals to really limit exposure to trypan blue and ensure that it's safely disposed. And this is everything from pipette tips to glass slides. Obviously, you need to ensure that all people handling this are properly trained. But ideally, what you should do is look for an alternative viability dye. And what are the alternatives? Well, just show you a couple of alternatives and their principles, and they're all based on the dye exclusion principle here. And basically, I've divided it into two categories, one being colorimetric dyes. In this category, we have the protein binding dye trypan blue, and we also have erythrocin that use bright field based cell counting, where you basically need to identify the cells based on their roundness and their size and how you do this. And this is a good method, but we also have alternatives. One alternative that we prefer is using fluorescent dyes. You have been introduced to the DNA and RNA binding fluorescent dye propydium iodide. There's another fluorescent dye that's DNA binding, that's DAPI. There's another one called 7AD. And more exotically, we perhaps have amino reactive dyes such as liva dye, fixable dyes that do this. And why do we prefer using fluorescent dyes and why we think it's a good solution? That is basically because they give an excellent signal to noise ratio. So in the picture below, we have used a color called acridine orange to label all cells. And in blue, you have DAPI, which labels the dead cells here. And this is very easy to distinguish what are cells and what are not cells in this case. So how do we do it at Chemomedic, and what we think is a good solution? That is basically, if you look at the top workflow from trypan blue based cell counting, you'd basically take a sample, you'd add your trypan blue dye, you'd mix the dye, you would load it into a chamber, you would count your cells, for example, manually or using an automated cell counter, and you would calculate and get your results. The analysis time here is typically minutes, whereas we in Contract have created a workflow that rolls around the via cassette, where you basically take your cell sample and you then aspirate a sample into the via 2 cassette, and then you run it in the instrument, in this case, in nucleocounter NC202. And the time frame for analysis here is seconds. Why do we think the via cassette is a good good solution? Well, basically for a variety of reasons. So when you looked at the picture I just showed you before, you see we really dramatically reduce the amount of steps you have to take in order to count a cell and get a viability assessment. And you've also integrated the sample, and we've done this by integrating the sample loading into the cassette. So basically you would press the top piston that would aspirate a sample from the bottom of the cassette into the fluidics chambers of the cassette, where it'll immediately come in contact with two fluorescent dyes, acridine orange, which is membrane permeable, meaning it'll stain total cells, living and dead cells. And as well, another dye is permeabilized inside there, or lyophilized inside the cassette, which is DAPI, and DAPI detects dead cells only. So basically, using these two fluorescent dyes, you're able to get your total cell count and your death cell count. What more with this Vi2 cassette, we've actually calibrated it, meaning that we've measured the volume in the imaging chamber for every individual cassette. So you're actually not just counting an area, you're actually counting a defined volume. Because we all know that if you've used a Bruker chamber, that the slide might go up and down a little bit. We actually take this into account using this cassette here. Also importantly, you have no exposure to the fluorescent dyes using the VIA cassette, because they're lyophilized inside the cassette and they cannot get out. So how does it work when you load it into the nucleocounter NC202? Well, the NCView NC200 software algorithm will basically make a specific detection of the total nuclei. This is shown in the top left corner with the green cells shown here. This is your total cell count. And in the top right corner, you'll have an image of the dead cells. And on that, you're able to merge this and you're able to identify which cells are alive and which ones are dead. So basically, from these pictures, you're able to get your viability reading and your total cell count within seconds. Importantly, you do not need to optimize it because it's intensity-based. So what is the good thing about using fluorescent dyes, such as acridine orange? Well, I said it labels cells, but it's actually labels nucleated cells. So what we see here is that we've taken PBMCs at roughly 2 million cells per ml, and we've spiked in increasing amounts of red blood cells. And then we've counted these samples on the nucleocounter NC202 using the Vi2 cassette. And at the same time, we've used an automated cell counter that's trypan blue based. And what we see here is that with increasing red blood cell concentration, you will actually get the same cell count, PBMC cell count, using the nucleocounter with a Vi2 principle. Whereas if you use a trypan blue based bright field cell counting, you will end up overestimating the amount of PBMCs you have in there. Really showing you one reason why we think it's a good idea to use acridine orange to specifically label nucleated cells. And this really brings me to the conclusion, which is that trypan blue is considered carcinogenic, mutagenic, and may affect reproduction. And as I showed in the beginning, trypan blue can cause swelling of dead cells. And the exposure to trypan blue can, over a prolonged time, cause cell death on its own or unspecific labeling. And really, colorimetric detection of cell viability may be error-prone in some cases. And this leads us to perhaps it's time to change and try something else. We really like the acridine orange DAPI approach because you really have an increased specificity of your cell detection and viability. Get this, and these dyes do not cause swelling or rupturing of your cells. And as I showed in the slide before, we're able to label PBMCs, but not red blood cells, or did not show the data, but also not beads. But I think another important aspect is really that the Vi2 cassette provides process simplification, really simplifying your cell counting workflow, but importantly, at the same time, it protects users from exposure to chemicals. And this is something that I personally like a lot when demoing this instrument. So with that, let's say thank you for attending our webinar and questions, and I guess we'll look at the poll now.
Yes, so exactly. I'm just going to share the results with you, Torsten. Thank you very much for your presentation and some great points on the potential issues if using trypan blue. So here are the results of the poll, and I thought maybe Torsten, you'd like to just go through them.
Well, I mean, we can see which viability did I use. A lot of people use trypan blue out there, and I guess that really shows that it is the traditional way of cell counting, and some of you use propylium iodide and DAPI and others. That's very interesting, and obviously, everybody, we're more than happy to discuss how to do this. And we see that we have a nice mixture of manual and automated cell count. This is actually split completely evenly, 50-50. And I guess this is a testament to how well the human eye actually is, that people are able to count cells manually. However, if you do begin to work in processes where more than one person is involved, automatic cell counting is traditionally preferred, simply because you reduce a lot of variation this way. And have anybody experienced cell viability decrease over time? Well, I guess this is kind of what I've expected, that many would see a viability decrease over time, and this is probably down to the way that you grow your cells or treat them, and you who do not see it is the same way. And I guess, will you be affected by the new EU legislation? Well, I don't know. That's a good question.
I like that so many of our participants don't know either. I think that's maybe something we need to follow up on with all of you and
exactly
and help you find out, because I think that is important.
We really appreciate your honesty.
Yes, for sure. So we'll now go to the Q&A session, and you can use, as I said, the little Q&A function at the bottom of the screen, and I see the questions are coming in now. So just letting you know that if your question isn't answered live here today, we will follow up with an email or a call and give you the answer. So you will have your question answered either way. But I'd like to start off. There was one question which was about, does the system work for bacterial cells? And I assume that was for the nucleocounter, and it doesn't. It works for insect cells, yeast cells, all sorts of mammalian cells, chicken embryo fibroblasts, blood cells, cells on microcarriers. You can continue the list, Torsten, I think, but it does not work for bacterial cells.
Exactly. And also, if you grow your primary cells, it'll also work well. So
yes,
you have that as well.
Yeah. So we'll start with one of the questions and I see people asking questions in the chat section. Please do so in the Q&A section, because that is the one I'll be monitoring from now on. So please move over there and put your questions in there. Thank you. So the first question I'd like to start or ask you, Torsten, is does diluting trypan blue in PBS help avoid excessive toxicity in the sample?
Well, PBS has its own issues. I mean, we know we call it physiologically buffered saline. However, we always recommend, if possible, to dilute your cells in trypan blue. We do this based on a publication that's out there that basically shows that diluting your cells in trypan blue will cause, diluting your cells in a PBS will cause them to rupture to some extent. So you'll really underestimate your total cell count. So for this reason, I would recommend not using PBS.
Thanks. And how does the toxicity of trypan blue apply to aggregated or cultured cells, in particular to MLRs?
Well, I mean, commenting on specific cell types is not something I would prefer to do, because most processes are different from customer to customer. That's at least what I see, that one time you have it one way, and it really goes down to how you grow your cells. But I would assume that you would get similar effects as shown in the slides here, that you will get some swelling of your cells, and maybe some of them will rupture.
So another question here is trypan blue uptake fluorescent under excitation in flow cytometry?
To my knowledge, trypan blue is mostly used using bright field microscopy. So that's what I would use.
I haven't heard of it used in flow either.
You would use a fluorescent dye.
And maybe a little bit of a related question, or at least staying within flow cytometry. What are some of the, or why are trypan blue values higher than fluorescent viability dyes in flow cytometry measurements?
Well, I mean, I guess this is what we're trying to explain here is that basically you will have trypan blue causing cellular swelling, and if you count this manually, I mean, these large aggregates are sometimes difficult to see, and you can also see that they might not be that strongly stained, and you might actually consider this not being a cell and discard it as debris. So if you do that to some proportion of your dead cells, you'd obviously get a lower total cell count, and you would get a higher cell viability. However, your live cell count would be unaffected by this, which is also what we've seen.
Thanks. One question here is, trypan blue is typically supplied in H20. Was the osmotic pressure causing cell death compared to using trypan blue resuspended in 1 times PBS?
Causing trypan blue. Okay, so this is a good question, and I will try to. I can't, honestly, I cannot remember the answer right now. So
that's fair.
Yeah.
This question is, how does the dilution of trypan blue with PBS alter stainability of cells and accurate live to dead cell discrimination?
So trypan blue, how will it change it, right? I mean, it depends on what dye you would use otherwise, right? I mean, I would say that with PBS in there, I mean, you will get a larger effect, because you will change the osmotic pressure in your media, basically. So I would say that you should you would probably get a slight underestimation of the amount of dead cells you have in there.
Thanks. And I think I have 1 or 2 more questions about trypan blue, and then there are quite a few questions about the nucleocounter and bio2 cassette system. So we'll move into that shortly. But how long do you need to expose trypan blue before the cells, or do you need to expose the cells to trypan blue before they start to change the morphology in morphology? I
mean, this is really down to the way you've treated your cells to begin with. So you can say they will not change morphology if you heat shock them or if you ethanol fix them or these things. So it's really down to the way that your cells die, how fast this effect will be, or if it even will occur. I guess this answers the question, that it's really down to how you treat your cells if you see this effect or not, because it's definitely not something you'll see for all cell types. Not all cell types, but not all ways that cells die.
Thanks. And one question here is, when is the new EU legislation, when will that be implemented? But I think it already has, right?
Yes. As far as, I mean, also important to note, we are not legal professionals in EU legislation or related, but this is, at least it's been put on this ECHA list of substances. So it is there, and as far as I know, it's also down to how every individual country applies the lists to their working environment.
Yes, and with EU legislation, there might be differences in when each country ratifies that legislation and puts it into effect. So that might also change a bit. So I'd recommend everybody to check their national pages for toxicity or drug toxicity and how to deal with them in the lab. So sorry, I was just getting caught up in a question. So if we move over to the nucleocounter system, I'll ask you a few questions on that, Torsten. So what is the minimal volume and cell number needed for detection on the Via2 cassette?
So the minimal volume, it's not philosophical, right? So the cassette will aspirate 60 microliters, and we are able to, with this instrument, count with a high precision and accuracy from around 50,000 cells per ml up to 10 million cells per ml. We and I always recommend that when you take a sample that you want to count with, you take at least 200 microliters. And the reason we do this is really not to do with the fact that we need 200 microliters, but more to do with the fact that we want to ensure that you take a representative sample of your cell suspension. And this is something that I've seen quite often, is that people get much higher precision, meaning low CVs of their cell count when they use larger volumes of cells to count.
Yeah, and does the system work for multinucleated cells like hepatocytes?
Yes, it may work, is the honest answer. So hepatocytes, as I see you have here, this we have gotten it to work with, yes. Yeah, so that is an algorithm that will fix this.
Yeah, thanks. What are the chances of cell aggregates clogging the capillaries of the system? So I guess these are the mixing channels in the Via2 cassette that are meant here.
That's a good question. So basically, we have a dedicated protocol for analyzing aggregated cells, and what this really does is we're able to dissociate the aggregates and count the cells individually, and this works really well. And for this reason, we do not have a clogging of the capillary systems.
And are there any cell culture media that interfere with the NC202 readings?
Any media that, well, to my knowledge, we haven't come across media that have had any deleterious effect to the cell counting performance of the Via2 cassette. However, there may be media out there that will affect it. I mean, I don't know.
But I mean, you do have a couple of years experience in traveling around and talking to customers. So I think you've talked to several hundreds, and if you haven't experienced it yet, then it doesn't seem like it would happen quite or very often. Now, so two technical questions. What is the shelf life of the cassettes? Do you remember? Because I do not.
Yes, and I should remember this. This is it's written on our website, first of all, and it is 1 year from receipt.
And then also, what is the price of the nucleo counter NC202? Or I have quite a few different questions about it, but of the system in general.
Honestly, I'm the technical expert here, and I do not know the prices of our cell counters. So
no, but I can maybe fill in a little bit and say that it does vary with the type of application that you're using, and there are some add-ons to add to the instrument. So it's really more of a discussion to have with one of our sales reps, and we're very happy for you to contact us so we can follow up and start some of those discussions, because it does depend a little bit on exactly how you're going to use the instrument and for what, and which additional software packages you may need or not. So we'll just do a few more questions, and I'll jump back into some of the Trypan Blue questions, because now I've had some more of those. You have mentioned a few times about Trypan Blue affecting fertility. Is there any way to detect this?
That I honestly do not know.
No. I guess there is literature
out there.
There
must be, right? I mean, there must be out there. There's a reason why companies have indicated to the European Chemical Agency that this is an issue. So there must have been done animal studies on it, is my guess. But how to detect this in us people, I do not know.
Okay, and so I'll just round off with one last question. And then we'll end the Q&A session after that. If the criterion is membrane permeability, why does it not work for bacteria? We can use PI for viability in bacteria.
That's a good question. I'm not an expert on bacteria, but what I can say is there are many different types of bacteria, and it seems that some bacteria will actually actively take up PI, and some will not, as I've seen. And this is basically only based on one review that had 4 pages on trying to discuss why some dyes worked in some cases and some didn't. I do not know. Maybe they actively uptake it. Maybe that could be.
Okay. Well, thank you very much, Torsten, for answering these questions. And as I said before, first of all, the session here is recorded and will be sent out to you. And the questions that haven't been answered, we will also follow up with you and answer those. If you have any other questions or want to contact us ever, please do so, sending us an email to contact@chemometic.com. And we also have other webinars available on our website that you can check out. And with that, I'd like to thank you very much, Torsten, for speaking to us today and answering questions. And also thank you very much to all of you joining, and have a great rest of the day. Goodbye.
Bye.